everyone. Can you hear me in the back? Good. Okay, cool. So my name's Max. I don't have, for the purposes of this talk, I don't have an affiliation. I have a day job, but it's not really related to this. So it's about creating genomes, the actual technical aspect of it. So right now, there's the first slide, but there's not much on it. We can just skip that. So right now, I know they're quoting numbers like 10 cents a base pair. I think in the next few years we can maybe hit 1 cent per base pair. That doesn't include assembly costs, but even at that level, it's 30 million per haploid genome. Plenty of room for improvements, I think. I hope. And so the first of these two, this two kind of project is Isothermal Amplification Array, which is basically if you have a library of every single 8-mer oligo, you can kind of chain those together and get larger sequences. So what we do is, you can see down here, so there's about 65,000 of them, which, you know, we can do that in microarrays, or we have done. Everyone remembers microarrays, I assume. So what you do is you get a single, you just synthesize a whole bunch of one strand, and it's got the nicking enzyme site in it, which like a restriction enzyme, but it only cuts one end. And from there you can add on an extra 8 base pairs onto the end of this thing, place those onto an array. Place it onto an array, and then you can do extension on it, probably, like with DNA microarrays. So you have each spot has a unique 8-mer on it, and then that's attached to this kind of foundation sequence with the nicking enzyme site on it. So we lay all those out, and then so what happens is you have put your DNTPs, your polymerase, your nicking enzyme. Polymerase comes in, synthesizes the second strand. Nickase chops it off, and then the polymerase, which is strand displacing, comes in and pushes that one off, generates a new one. The cycle repeats itself until you have a pool of 8-mers floating around. And you can pull those off and do other stuff with those. They do need to be phosphorylated or not, as that will come up later. You can use those. They won't participate in a further ligation reaction, but they can use to guide the extension of other strands. So we take those oligos and we transfer them into ligation. So it's based on paper, which was, I thought, quite good. Basically, you just have one 8-mer kind of immobilized on a surface, and then if you feed in other 8-mers, as long as they have they're fully hybridized, a ligase will come in and ligate them together, and you can do this and kind of extend it ladder-like. And then it's pretty nice. It has, you know, if you make the reaction stringent enough, you can get pretty accurate base pairing. You know, if there's for some reason you have a shorter oligo or one with an error, it might not necessarily land on there. It'll get outcompeted by the correct oligo, and you can kind of extend this further and further and get some length of fragment. We don't know at the moment. So when they tried this in the original paper, this is just like wild type T4 ligase and just regular reaction conditions. They got about 128 base pairs pretty reliably, and this is double-stranded DNA, which is nice because it doesn't fold back in on itself and all that stuff. So we don't know at the moment how long we can make it, but it's worth looking at. And then down at the bottom, you can see this will come up later. If you have guide strands in there, aren't phosphorylated, they can still be used to guide the ligase because it needs that certain amount of double-strandedness to do the ligation. And you can generate single-stranded overhangs using that. Let's see. So this is in use, at least the nicking and polymerase thing. I think it was a guy from George Church's lab at some point was doing. It's the same kind of thing, just kind of, this is with much longer oligos. So it's just kind of to amplify traditional phosphoramidite synthesis. You have more oligo to work with, and then here they're doing like overlap extension PCR, pretty traditional stuff. With the library, the 65,000-ish oligos minus ones that are like self-complementary, you at least have them all on hand. You just need to add the enzymes, the DNTPs, and just wait, and you can do that parallel if you want to. And then you have them all on hand, and to some degree, yeah, if you have a short oligo that will like perfectly match with itself, you can't use that one, but you can algorithmically determine the best way to assemble a sequence based off of that and try to get as long of a sequence as you can until you get a sequence that's not possible to be made with this. And there's room for traditional synthesis as well to bridge the gaps. So that's fun. And then the second one is Recombinase-mediated Assembly. So I should point out, everyone knows recombinases. It's usually site-specific kind of Krelok stuff, but this enzyme, UVSX, it's related to Rec-A, if everyone knows Rec-A, or like the RAD enzymes from yeast and humans. What they do is they're used in double-stranded break repair, but they will bind to a short sequence of single-stranded DNA and then kind of scan through double-stranded, other double-stranded DNA that's around. And then when they find a match, a very close match, they will displace the third strand on that double-stranded DNA and kind of shove in the single-stranded DNA. So here it's used for like isothermal PCR. You don't need to temperature cycle. You just have your amplicon here. The X, which is those yellow blobs, binds on the primer, and it just shoves the primer into the end of the amplicon, and then polymerase is able to access this now underhang and then synthesize a new strand, and there's a PDB image of it. So it's pretty cool. People are using it mostly for this kind of isothermal PCR stuff for low resource environments. You can do PCR without a PCR machine and then get enough amplicon to detect. And what we do instead is you have a fragment. Like I mentioned, there's the underhang on it. We don't know exactly how long these fragments are going to be, but it should be fairly flexible on that end. So we have the female and the male end, just for clarity's sake. So you have the X enzyme in there, coats the single-stranded overhang, and then it scans through. I'm not showing the scanning part, but it scans through all the double-stranded DNA in the reaction. When it finds the match, so that overhang has to match the female end of the other fragment, it places it in, displaces the third strand, and then ligase will hopefully ligate that at that end, and during that time or shortly thereafter, the exonuclease will come in, degrade that extra third strand that we have here, and then the ligase will come in and ligate that second strand, and then this now larger strand is probably also simultaneously participating in this kind of homology search reaction, and you eventually just throw them all in there and you get long stretch of DNA all fully assembled. So there's a lot of questions on making these enzymes work well, making sure all these parts work correctly, but there's a lot of room for improvement in the enzymes and the stoichiometry of the reaction. People are basically just using the wild type enzyme for a lot of these, and it works pretty well in other applications anyway. So this is, as I mentioned, it's Rec-A, so it's very similar to the double-stranded break repair. You have a break. Other enzymes chew back the sequence on the break, and then X, or in this case Rec-A, not shown here, will scan through the sister DNA and then find a match, and then it just does homologous recombination basically. So people have used Rec-A in DNA assembly in general, but it's basically just an improved tweak to Gibson. It's single strand to single strand, which these enzymes don't really do. They work much better scanning through double-stranded DNA. We haven't really figured that out yet, but we can still work with them. But this, basically it's right now with in vitro assembly. There's a few different methods. Everyone knows, Gibson, stuff like that, but you get a lot of weird side products that you can't easily separate out a lot of the time. You have scars and seams from restriction sites. You have a lot of antibiotic resistant genes you got to shove in there, which is kind of a pain. And then for pretty much all of them, you're relying on just passive base pairing, which is not that great. Well, I'm basically done. Sorry, I didn't see that sign. And then in vivo has its own problems. You just hope for the best with the cell. Anyway, I'll take questions. Anyway, that's all. Thank you. It was a lot to get through, but I figured best to just push through. Questions from the audience, from the cafe? Anyone? Okay. Oh, yes. Omri.
The first slide, you said the cost per base pair for phosphoramidite is capped at 0.01 cents a base pair.
Well, I would like to ask you about that because I hear you're at Twist
It can
now.
go way down, so just...
Okay, well, I'll believe it when I see it. Been promising for a long time. Anyway, any other questions? Yes, Jeff?
Have you done calculations on the impact of mispaired ligation, and how frequently you're going to have an error, and how big of a problem this is going to be to really implement the 128-mer synthesis?
It's going to be interesting, I will say. I mean, yeah, we can make it much more stringent. Probably engineering the ligase would be definitely parts of this project. It's not just kind of plug and play with the oligos and the ligase. But, you make it very harsh conditions, make sure only the strong base pairing survives or whatever, and then see what happens. There's a lot of engineering work to be done, but worth investigating, I think.
So, quick question about the 8-mers over here.
Hi.
So, when you make the initial 8-mers, do you have ideas about how to make those very high fidelity in the first instance?
It's tough. So, I mean, the first 4 bases you can pretty much do. I mean, that's only 256 oligos. You can have the first 6 bases on there and then kind of dot those out. But I'm not sure the efficiency of that kind of, it's like UV masking, what they do with DNA microarrays. I'm not sure the efficiency on that. To some degree, it's error correcting because a short oligo or whatever won't get, or hopefully won't get incorporated as efficiently. But I'll still have to look into that. Yes, it's an ongoing question. Yes. I make proteins for a living. It's a company, I don't know if I should really mention them, it's not at all related. This is kind of my third, it's not even like my side stuff, it's just, this is like free time after free time kind of thing. Do we have time for more? Sorry, okay, sorry. Come find me later. Okay. I'll be around.